Welcome back to lesson number 2, genetic recombination and gene linkage. So genetic recombination is a word used to describe the kind of combination that happens after there's been a separation between the alleles. So proteins are large complex polymers essential to life that provide structure and provide structure for tissues and organs. So the summary is protein help the cells, the tissues, organs to grow properly. So the new combination of gene produced by crossing over an independent assortment is called recombination. So as the name implies, a recombination, that is to say, a combination after the breaking up, after the separation. So a combination after the separation is called recombination. So, and as the name, combinations of genes due to independent assortment can be calculated using the formula 2 raised to the power of n. So where n is the number of chromosome pairs. So let's say, for example, the humans that have 23 chromosomes. So it's going to be 2 raised to the power of 23. And that's going to give you the number of possible alleles to be expected from that individual which has 23 chromosomes. So any possible male can fertilize any possible female gamete. So the possible combinations after the fertilization will be 2 raised to the power of 23 times 2 raised to the power of 23. So that is to say the possible alleles which is expected from a person after fertilization is going to be 2 raised to the power of 46. So genetic recombination helps to increase variation because you're having combination from different alleles, and that would help to bring different genes together. Genes located to each other on the same chromosomes are said to be linked. So when there is a connection, when genes are located close to each other on the same chromosome, they're said to be linked. For example, gene A and B, uppercase, are linked. Gene A and B, lowercase, are also linked. So they usually travel together during gamete formation. If you remember, the law of Mendel says there is a law of independent assortment, that the genes, the alleles, do not have a unique pathway in which to get inherited, but this law has been affected by this principle that says genes that are linked together get inherited together. So gene linkage results in an exception to Mendel's law of independent assortment. So you can see we've got replicated homologous chromosomes here. So these homologous chromosomes, they will separate. So it's separated into 2 different cells. So during meiosis 1, this is what you have, mind you, because as you know, in meiosis there is haploid, so they have to split to form a what? To form a parental. So now, as you can see, so the centromeres separate and a gamete are formed. So these are the gametes that are formed from each parent. So one gamete from one parent, the other gamete from the other parent, they'll fuse together to form a fetus. That's the reason why we say we have 46 daughter cells from meiosis. So, and chromosome map is something very important. So chromosome map it helps to link how far genes are away from each other on a chromosome. So crossing over data can be used to create a chromosome map. So the pictures of how genes are arranged on the chromosome can be used to create a chromosome map. So it's something which is pretty easy to create a chromosome map, but I think you should try to pay attention to trying to draw at least one chromosome map. Let me try to do one for you. Let's say, for example, the distance between gene Y, W, V, M, and R. So let's create a case study with these genes we have here. So let's say we want to construct a chromosome map between the gene Y, just give me 1 minute, please. The gene Y, the gene W, V, and M. Now let's say gene Y to M is equals to 25%, for example. So genes Y to V is 20%. Then gene Y to V. So let's say gene W to V is 5%. Then gene W to M would be what? So 25, 20. W to M would be, it's going to give you, so W to M is going to give you something like 10, let's see. So let's try to draw it. So we have Y, we have W, we have V, and we have M. So I said V to M is 25. That's 25. Okay. So and I said Y to V is 20. So if Y to V is 20, then V to M will be 5. So this is going to be 5. Then if W to V is also 5, so that means 10 out of 25. So then Y to W would be 15. So that is to say the gap between Y and W is more than the gap between W and V. So this everything is supposed to give you 25 if we were correct. 5, that's 25, yeah. So this is how to construct the map, you can see. So you can make it cleaner by putting 15 here. Then this put a V, sorry. Put a 5. So 15, 5, 5. So this is how to construct the chromosome map. It's not something difficult. Or let's construct another one. Let's use ABC. So let's say, for example, A, B, C, D. A to D is 25%. And A to B is 30%. C to D is 15%. And B to D is 5%. B to C is 20%. So this is a bit longer. This is in your worksheet. You can try to study it yourself. So what you're gonna do is you just create create a space, then B, create a space, C, create a space, and D. But this cannot happen because A to B is 30. So that means we will put a B here and we'll put a D here. I'm gonna put a C here. So because the space between them has to be the yardstick. So A to C, and to D. So now it says A to D is 25. So this is 25. And A to B is 30. So let's start with the shortest. C to D is 15. You write 15 here. And let me write the numbers with another pen to make it easier. So let's put 15 here. Then B to D, this one here is 5. So then A to C, what is A to C? He didn't say A to C. But since we can know that all of it, A to B is 30, so we would be expecting this, all of this to be 30. So we've got 15, it is 20, so this is going to be 10. So now that means that the distance between gene C and D is more than the distance between gene D and B. So that is to say gene D and B would most likely be inherited together than genes C and D. So, and also gene A and B would most likely not get inherited together because they're too far away from each other. So this is the principle of chromosome map, which I think is pretty easy for everybody to understand. So now let's move on to the next one. So polyploidy is an occurrence of one or more extra sets of all chromosomes in an organism. As you know, the karyotype shows that genes are arranged in pairs like this. So it always arranged in pairs, but sometimes when there's polyploidy, there could be something like this. There could also be something like this. So all of these are polyploidy. So triploidy organisms would have 3N instead of 2N, which means that it has 3 complete sets of chromosomes. Many agricultural crops are polyploid, wheat, for example, oats, sugarcane. So polyploid plants often have increased vigor and size. So they are usually large in size, and also they have better quality than the other ones. So this is all we have for this short chapter. It's not that long. So which is not true about polyploidy. It is caused by crossing over. It can produce a hardy and vigorous plant. It never occurs in animals. It's lethal in humans. So which of this is not true? Is polyploidy caused by crossing over? So that's the question. Is it caused by crossing over? So most likely it's not caused by crossing over. So polyploidy, it happens, it says it's not true about it. It is lethal in humans. It can produce hardy, vigorous plants. So the answer is most likely going to be the, the so as I was saying, so the answer would be it's caused by crossing over. It's not caused by crossing over, which does not contribute to genetic variation here. Which of these does not contribute to genetic variation? So the crossing over contributes to it. Meiosis does, random mating. So chromosome number does not contribute to it. So this question here, so the answer, it says the housefly has 6 pairs of chromosomes, so it's going to be 2 raised to the power of 6. And I think this is the answer. You can use a calculator. So based on the chromosome map shown, which two alleles have the least frequency of crossover? That is to say, which of them would not most likely cross over? Because which of them would not most likely cross over? They have the least frequency of crossing over. If you take a look, you will see this and this will not cross over. Why? Because they are linked together. So genes are linked together, do not cross over. So the ones that are linked together are inherited together, but they will not, most likely not cross over. R and Y would cross over because they're far away from each other. R and W can also cross over because they far from each other. Y and M can also cross over, they far from each other, but these two are too close. They will rather be inherited together. Okay, that marks the end of the chapter 2. So I wish you all the best in your exam.